Ready to go, folks. Okay, today's speaker is Don Smith, and Don is a professor, distinguished professor, in the Department of Microbiology and Environmental Toxicology. Don has had a superlative education, getting a bachelor degree at the University of California at Santa Cruz, going on to get a master's at Cal State San Francisco, and then getting a PhD at the University of California at Santa Cruz. It doesn't get any better than that. All right. And Don did postdoc at Maryland, postdoc at UCSF, and then was hired here as a faculty member in '96. So one thing I want to say about Don is he's won a number of awards, but just last year, every year the science division picks one faculty member as their outstanding faculty member of the year, and that's just based on research, teaching, and service. And Don has been superb at all of those, and Don was the winner for last year. So, Don Smith.
Can people hear me okay? Is this too loud? That's right? Okay. I want to first thank everybody for coming this morning. It's a beautiful spring morning here, and you're taking time out of what you normally would be doing to come and listen to what I have to say, and I greatly appreciate that. And I want to thank Barry for that really gracious introduction. What I want to talk to you about today represents about the last 12 years or so of research in my lab here at UC Santa Cruz. And I'll just give you a little background. I started out actually as a marine biologist, which is why I went to Moss Landing Marine Labs through San Francisco State University. And I was actually a marine biology major when I was an undergraduate at a couple different schools, most notably UC Santa Cruz. And partway through my work in marine biology, I realized that I wanted to do more that was contributing to improving the quality of our environment, particularly as it affected the animals, wild animals in our environment, but also I became more interested in actually doing work that would contribute to improving human health. And I was always interested in toxicology, in particular, how we might be exposed to different kinds of toxic metals like lead and things, and how that would affect our health and our development. And that put me on a trajectory to do the work that I'm going to talk about today. And so you can see the title here is work we've been doing in looking at the environmental contributors to attention deficit disorders, and try to establish the causality, meaning how do we understand what is causing these kinds of disorders, and what kind of therapeutic approaches might be used to treat the symptoms of those disorders. Here's an outline of what I'm going to talk to you about. Attention deficit ADHD, I'm sure all of you have heard of that. We'll talk a little bit about the symptoms, underlying neurobiology, and also some of the therapies that are used clinically for this. And then I'm going to get into the work we've been doing looking at developmental exposure to the trace metal manganese, and I'll talk a lot about that. And I'm going to demonstrate to you that developmental exposure to manganese is now we've established can cause ADHD-like symptoms in an animal model of attention deficit. Now that we have that animal model, we've explored various therapies to treat the symptoms in that animal model for the attention deficits. And then, of course, since it's an animal model, we can go a long ways to trying to understand and using this model to understand what the neurobiological causes are of those symptoms that are expressed in the animal model. So the first problem I want to introduce is attention deficit hyperactivity disorder. All of you have heard of this, I'm sure. It is the most prevalent behavioral disorder in children globally. Upwards of 5 to 7% of kids under the age of 18 have been diagnosed with ADHD. 2 to 3% of those kids go on to retain the symptoms of ADHD into adulthood. The characterization of the symptoms, as many of us might be familiar with, are deficits in attention or inattention, impulsivity and hyperactivity, emotional dysregulation, which is something that we don't often think about with ADHD, psychomotor deficits, fine motor coordination deficits, and behavioral self-monitoring deficits. And I'll talk quite a bit about that in a minute. What I think is really important to emphasize here, as the most predominant neurobehavioral disorder in children, it comes with significant long-term impacts on the quality of life of people who are diagnosed with ADHD. It affects their ability to establish personal relationships. It affects their academic success. It increases the likelihood that they'll develop substance abuse disorders, has a number of different adverse occupational outcomes and how they're able to actually enter an occupation and support themselves. And in some cases, it can even increase the probability of criminal behavior. Right? And there's, like all behavioral disorders, and in fact, all medical conditions, there's a significant economic burden and cost that we spend in the United States and globally in treating the symptoms in the children and the young adults who have ADHD. And in the United States, it's estimated that it's more than $250 billion a year just in managing the diagnosed cases of ADHD. So it's a huge problem. Now, ADHD as a behavioral disorder is what's referred to as a classic multifactorial disorder. And that means that there are recognized multiple factors that may contribute to increasing the risk of being diagnosed with ADHD. But the actual cause and how each of those individual factors may contribute to it is actually relatively poorly understood. The ADHD symptoms, it is recognized, they're believed to arise or occur largely because of deficits in certain neurotransmitter systems in the prefrontal cortex of the brain. That mainly involves deficits in dopamine function and norepinephrine function. And they refer to that as a hypofunctioning condition that is generally recognized to occur and maybe underlie the ADHD symptoms. And fortunately, in many cases, there are a number of different therapies, medications that have been proven to be generally effective in treating the ADHD symptoms. They don't, of course, treat the disorder itself, they just treat the symptoms of the disorder. And the most commonly prescribed one is Ritalin, which is otherwise known as methylphenidate, and I'm going to talk about that quite a bit today. And Ritalin is actually a stimulant drug, meaning that it increases levels of dopamine in the extracellular space around our brain cells, and they believe that that increased levels of dopamine is what leads to the benefit of Ritalin or methylphenidate as a therapy. So the second problem that I want to introduce has to do with early life or developmental manganese exposure. And I show on this slide a number of different epidemiological studies that have been conducted over the past 12 to 15 years or so. And those studies have collectively shown that there's an increased risk of ADHD-like symptoms with elevated manganese exposure, particularly if the elevated exposure occurred during early childhood development. And those kinds of risks have been shown to be in one study, which is probably the most powerful and compelling done to date, in a large cohort of kids in Denmark, that elevated exposure to manganese in drinking water, even slight elevations during neurodevelopment in childhood, was associated with a 50% increased risk of the inattentive subtype of ADHD. Other studies have shown that elevated manganese exposure increases inattention and impulsivity, hyperactivity. It is associated with reduced IQ, either the performance or the verbal or the full-scale IQ measures that have been made. That the increased exposure to manganese is associated with deficits in psychomotor function. All these studies collectively are showing that there is significant reason to be concerned about how kids may be exposed and suffering the consequences due to elevated manganese exposure. But one point I want to make in showing these epidemiological studies is studies like these done in humans can only establish a statistical association between an exposure, like manganese in this case, and the adverse health outcome. They cannot establish that the exposure is causing the health outcome, the adverse health outcome, only that there's a statistical association. And that's an important point. Now let me introduce manganese. So I've been studying metals and trace metals for almost my entire career, mostly focusing on ones that are toxic. But manganese is relatively unusual for a toxicologist in that it's shown up here circled red in a transition series of trace metals. And it is a biologically essential metal. That means that we require it in our diet for normal biological function. But like all things that we encounter in our environment, and I refer to it sort of like the Goldilocks challenge here, that we need a certain amount in our diet to maintain normal biological function. If we don't get enough of it in our diet, we suffer deficiencies, and there's health effects due to that. But if we get too much, we suffer toxicity, and there's adverse health effects caused by that. And this is often represented in a sort of a U-shaped dose response curve that I show on the right side of this slide. And you can see the red rectangle at the bottom of that. That's the levels that we might need in our body for normal biological function. Too little is deficiency, too much it's toxicity, and our systems are designed to try to maintain it right in that sweet spot. Not too much, not too little. Okay? Now the problem with children, particularly very young children, is they don't metabolize manganese as effectively as we do as adults. Children are known to absorb much more manganese that they take in orally or through the respiratory tract than we do as adults. And of that amount of manganese that they take in, they're known to withhold and retain it at much higher levels than we do as adults. And that's because the main physiological way that we manage manganese as adults is we take it in our diet. Even if we take in too much, we excrete the excess via our liver back into our bile, and it never hits our bloodstream, our main bloodstream. So we're able to regulate it pretty well. And in fact, it's very difficult for adults to be overexposed to manganese unless you have an occupation like a welder or a manganese miner or something like that, where you're being exposed to high levels via inhalation. But for most adults, it's very difficult to get overexposed to manganese. But that's not the case for children. And it's because they take in and absorb and retain much more than per amount of manganese they might be exposed to than adults. And on this slide, I make that point that they're more susceptible because of just their metabolism, but they're also more susceptible because infants and young children are undergoing very rapid and important neurodevelopmental processes in their brains as their brains are developing, which really starts for humans in the third trimester of pregnancy, and it continues on up through adolescence. The prefrontal cortex of the brain, which is one of the areas that I'm going to talk about today, continues to develop in humans up through their early 20s. And it's during that period of development that they're most susceptible to altering that development if there's some perturbation or insult, like exposure to manganese. So where do we get manganese that might be too high? Well, in the United States, it's estimated that 7% of municipal drinking water wells have naturally elevated levels of manganese. That's one very important source. Also, this is also particularly relevant in Santa Cruz, infant-based formulas, and especially soy-based infant formulas, contain manganese levels that are naturally high and about 50 to 500 times higher than in human breast milk. So children that are receiving, particularly being fed largely infant formulas, even bovine cow-based formulas, but especially soy-based formulas, if they're lactose intolerant, are getting tens to hundreds of times more manganese in their diet than they would normally get from human breast milk. And they're not able to regulate that to the same extent as adults, like I mentioned. And a third important source of exposure environmentally could come from different industrial processes. And in the bottom of this slide, I show a couple of pictures of these ferromanganese alloy plants. We've also been involved in studies in a cohort of Italian children in the north part of Italy, where they have these ferromanganese alloy plants that have been in operation for decades to over 100 years. And the communities where these ferroalloy plants exist have been contaminated with the emissions from these plants that fall out and expose the people in the area. And we've been studying those kids for over 10 years now. Okay, so I've shared with you the two problems that I think are important. One is that ADHD is prevalent and the causes of it are not well understood, particularly the environmental causes of it. And two, exposure to manganese, particularly for vulnerable populations like young children, is potentially a significant health threat. And so knowing that, we embarked upon a series of studies, probably about 10 years ago now, where we actually wanted to establish the causal relationship between exposure to manganese during development and whether it could actually cause ADHD-like symptoms. And if naturally we can't do that in humans, even though we were involved in some human studies, so we wanted to develop an animal model that we thought could recapitulate the ADHD-like symptoms that have been shown in kids. And then if we were successful in doing that, which we were, and I'll show you, then we could use that animal model to evaluate different therapies to see how effective they were at treating those symptoms. And we could also use that animal model then to ask questions about what is it that's happening in the brain of these animals that is contributing to those symptoms that would help shed light on what is causing the symptoms that occur in ADHD-diagnosed children. So I'm going to start on and tell you this kind of work we've done. So this is just a graphic that's meant to show sort of the overall study design. And we do this in rats, and I'll show you what they look like in a second. But in this model, we take these rats and we expose them orally every day to a little bit of manganese that's comparable to what children would be getting from soy-based infant formula or contaminated well water. And we expose them orally every day only for the first 3 weeks of their life, up until when they're weaned from their mothers. Okay? And that first 3 weeks of their life corresponds to maybe the first couple of years of a child's life. Then we train the animals in a series of these behavioral tasks that have been developed to mimic the kinds of tests that are used in diagnosing ADHD diagnosed children. And then after those tasks, we can measure a number of different things in the animal's brains to try to understand what the neurobiological alterations may be. Now, let me explain to you these tasks, because I think they're pretty interesting, and they're rather clever. So in the middle of this slide, I show a picture of this behavioral chamber that we use, and it's referred to as a 5-choice serial reaction time chamber. You don't need to remember that. On these chambers, we have rats that have been exposed early in life to either manganese or just to water. And then we train them through a series of tasks in these chambers, and we train them to respond to a visual cue that's presented in one of our 5 response ports. I don't know if you can see my cursor, but here's a diagram of the inside, and here are these response ports on the left side. And on the back of it is a food magazine port. And so these rats, and they're actually remarkably smart, these rats are trained over a series of weeks to sit in this chamber. They're there for an hour, and when the light comes on for only a second or a fraction of a second, they're trained to pay attention for where the light's going to come on at which response port. And when the light comes on, they poke their nose in the port, and by poking their nose, that breaks a photo beam, and if it's the correct choice where the light was presented, then they're given a food pellet reward. And that process, waiting for the light's illuminated, they make a response. If it's the right one, they get a food pellet reward. That would be referred to as one trial. And these animals undergo 150 trials in 50 minutes. And so the value of doing this kind of work in an animal model is that you can do things like train them extensively that you could never do with a child in a clinical setting. Right? And so we train them in this operant chamber to wait for the light, and we can make it very challenging by having them turn around and wait, and then making them wait 3 to 5 to 6 seconds, which is a long time for a rat, before the light's presented. And they can do a number of things. They can get distracted, stop paying attention. They can miss the cue because it's only on for a fraction of a second. They can respond to the port before the light's even presented because they get tired waiting, which would be a measure of impulse control or lack of impulse control. So we train them in a series of those tasks, and then we train them in a subsequent task that's even more challenging. In the subsequent task, we have 9 different odorants in jars, and this is looking at the back of this apparatus, and these odor jars are all connected by tubes to one of those different response ports. And it's all computer controlled, and we can activate that before the light is presented that the animal's waiting for. We can present the smell of a cue, like strawberry or orange or cinnamon. And these odor distractors are extremely distracting to the rats, because rats, of course, they rely heavily on olfaction for much of what they do. And we can test the extent to which these rats can be easily distracted by that odor and then miss the visual cue and not get the food reward. And so the rats learn over time that the odor is a distracting thing, and they may learn over time to ignore it because it's an irrelevant cue, and to pay more attention to the port where the light's going to come on because that's the relevant cue. And that's exactly what they do in children when they do tests like this clinically, is they present visual distractors or auditory distractors, and to see how well the children can maintain a focus on the task at hand for whatever the salient cue is they're waiting for, and ignore the irrelevant non-salient cues. That's a classic approach that people use. So we did a series of studies with these rats using these operant chambers where we could test their attentional focus and their ability to withhold responding inappropriately, their impulse control, their distractibility, and so on. And we recognized too that that because many ADHD children are also diagnosed with psychomotor deficits, fine motor coordination deficits, we also wanted to evaluate whether we could test how well manganese or whether manganese affected the animals' sensory motor deficits. And for that, we used a different apparatus that's shown on the bottom here. And you see this rat in a little clear box. And box is referred to as the Montoya staircase task. Doesn't matter if you remember that. But on this task, the animal is trained to sit in this little clear box, and there's a descending staircase of wells on the right and left side of the animal where the animal's laying. And on those different platforms of steps are different color-coded food pellets. And so the animal can easily reach the ones at the very top steps quite easily. But for the ones that are down below, they have to reach down and feel with their paws and manipulate and grab a pellet to retrieve it to eat it. And so we can test to the extent to which the animals are actually effectively grasping and retrieving and eating the pellets, or whether they're dropping them and they end up on a different step. And that's a measure of how well the animals are able to express their sensory motor function. Okay, so these are the main tasks we use. And I want to emphasize the amount of work, and this is something that I think we're all very proud of here at UC Santa Cruz. The series of tasks that I show here, it's a typical training series for these animals, takes maybe upwards of 100 days, over 3 months. And this is 3 months of testing where the animals are tested 6 days a week for 3 months solid, and it's 8 hours a day. So we have a large study, maybe 120 or 130 animals. Each of them, we can test 16 at a time, and it takes about an hour. So we have a small army of undergraduate student research volunteers that come in starting at 8 o'clock in the morning, and they'll work 2 hours, 6 days a week for 3 months, and we'll have a new shift come in at the next hour, and another shift for the next hour, and we finally finish at 6 o'clock in the evening. And we're testing these animals. And if it wasn't for the fact that we had these undergraduate research volunteers who have been just incredibly committed and engaged and excited, we wouldn't have been able to do this work at all, because it would have been just way too labor intensive. Okay, so here's a video just so you get an idea of that. I put my GoPro camera inside one of these testing chambers, and I want to show you what this animal is actually doing. You can see here that the animal, in this case, it's a pigmented rat. It's a Long Evans strain of rat. It's got both black and white fur. And the pigmented rats, as opposed to albinos, are important for these kinds of tasks because they need the pigmentation in the eyes for the increased visual acuity that's required for them to perform well in this task. So here you see the animal shown. It's facing the response wall, and in behind it, to your left, is the pellet dispenser wall where it's going to get its reward if it gets it right. And you can see the animal's waiting, and you'll see a light comes on. He pokes his nose to the right place, turns around, gets his food pellet reward, turns around, gets ready for the next trial. And now he's having a hard time waiting, so he responds before the light comes on, and that's referred to as a premature response. And when the animal makes a mistake like that, the lights on the whole chamber go out, and he sits in the dark for 10 seconds. So it's like a timeout that you might give, a misbehaving toddler. But they learn that over time that this is a kind of behavior that they're not getting a reward, but they also have all the cues that they made a mistake because the lights go out and they have to wait. So again, that was one trial, and they do 150 of those a day, 6 days a week for 3 months. So they're extremely well trained, very smart. We're proud of all of them. Okay, so now let me, I've done the whole introduction. Let me show you some of the things we found. So I mentioned to you in the beginning that ADHD children often exhibit something that's referred to as impaired behavioral error monitoring. And what that means is when a child makes a mistake, and this is not just a child, but anybody, when you make a mistake at some task and it doesn't work out, you don't get the expected outcome of the task that you were expecting, we are all aware of making that mistake generally, and we adjust our behavior in subsequent efforts to increase the probability of success at whatever our task is. So we're monitoring our performance, our behavior, and we do this in everything we do. Oftentimes we don't realize it, but we do it in most everything we do. Clinical studies have shown that children diagnosed with ADHD actually have deficits in error monitoring. And that means that if they make a mistake, it doesn't bother them so much. And so they don't learn and they're less able to make adjustments, behavioral adjustments to improve their performance on tasks that they're engaged in, okay? And they're also shown, ADHD children, to be more sensitive to rewards. But not just any kind of reward, but more sensitive to immediate rewards and less sensitive to longer-term rewards. There's a classic behavioral paradigm that they use in clinical settings where a child will be performing some task on a computer as part of their assessment. And in this task, they're given a choice of going for the immediate smaller reward or going for the longer, more potentially distant in the task, a larger reward. And most people would be like weighing out the consequences. I can go for the small reward now, or I can wait and go for the bigger reward later. Most people oftentimes will defer and go for the bigger reward. But children diagnosed with ADHD are more sensitive to that immediate reward that comes closer in time to when they're performing a particular task. And so let me show you what our rats have done. So in this task, and I'm showing you a figure here, in this task, we're looking at whether an animal got a correct response into the lighted port, but we're measuring the proportion of time they got the correct response in the lighted port, depending on how they did in the prior trial, whether they got it right or wrong. Now, most of us, if we make a mistake on something, we're aware of that mistake and we do some adjustments on our subsequent actions to improve our performance. And sometimes that mistake, depending on what the consequence was, it can be so upsetting to us that it'll actually affect our performance on subsequent tasks that are occurring near time. And we see here, if I can show you with my cursor, the animals that didn't get manganese, which are the open white circles, you can see if on trials where they got the prior trial correct, they do pretty well. They got over 60% of their trials correct. And on trials where they made a mistake on the prior trial, their number of correct trials dropped. They were negatively affected. Their performance was negatively affected by them making a mistake in the prior trial. Okay? So they're aware that they made a mistake in the prior trial. But the animals who were exposed to manganese just during the first 3 weeks of their life, they not only performed more poorly compared to the control animals, which is this difference, but they showed no reactivity or change in performance regardless of whether they made a mistake on the prior trial or not. And so that's evidence to us in this animal model that they have a deficit in error monitoring, just like ADHD children have been shown to do. And I also mentioned ADHD children are more sensitive to immediate rewards. And so we added a different component to this task. And that's why I'm showing this figure on the right. And on this task, we not only measured how well the animal performed based on how well they did on the prior trial, whether they got it right or wrong, but then on the trial that they got right, we also withheld their food reward on a subset of those trials. The food reward is the whole reason they're waiting to make that choice to get that food reward. And so the control animals are in the dark bars on the left, and we see that their correct response performance, again, is about over 60%, like it was in the prior task. When they make an error in the prior trial, it drops. They're affected by making that mistake on the prior trial. But if we withhold a food reward from them when they got the task correct, the trial correct, it didn't bother them at all. It didn't affect their performance at all. But for the manganese rats, they were not only not affected by making a mistake in their prior trial, but they were significantly affected if they didn't get their food reward when they got it right. So this recapitulates, again, what we've seen, what's been reported in ADHD children, again, that they have a hard time monitoring their performance, and if they make mistakes and making adjustments, and they're much more sensitive and responsive to immediate rewards as opposed to longer-term rewards. So I also mentioned to you that ADHD children often exhibit impaired coordination and psychomotor function, and that Ritalin, I didn't mention this, but Ritalin, or methylphenidate, has been shown to improve those symptoms in many, not all, but in many children who have those coordination problems. So in these rats that are in this Montoya staircase reaching down for the food pellets, on the steps that are really close to the rat, they can reach them, both the control and manganese rats can reach them equally well and the total number of pellets they have on those very shallow steps is 6, so they're able to get all of them and eat them. But when you look at the farther down, more difficult to reach steps, the control animals are retrieving and taking fewer pellets, but the manganese animals are doing even worse. So that's the deficit the manganese animals have in their sensory motor function, this difference here and here. On the x-axis, I have not only step level, the shallow and the deep steps, but also the dose of Ritalin that they got. And we gave these animals Ritalin orally on a little piece of cookie right before they went into their testing chamber every day. And we see for the animals that got the higher dose of Ritalin or methylphenidate, this is the highest dose, that totally rescued their sensory motor deficits. And so the highest dose animals, manganese animals are exposed to the highest dose, are now performing just like the controls who got the highest dose, or just like the controls who didn't get any methylphenidate or Ritalin. Right? So this recapitulates to a fair degree what is also reported clinically, that children who do show these coordination deficits that are comorbid with ADHD diagnosis oftentimes do respond favorably to medications like Ritalin. Okay, so the last thing in terms of behavioral data I want to show you, it relates to attentional function, which is, of course, one of the main features of children who are diagnosed with ADHD. And when we think of attentional function, there's various kinds of attention that we might think about. There's the attention where you have to sort of maintain attentional awareness at the stimulus or whatever it is that you're waiting for in front of you, right? And so right now, you're maintaining, hopefully, a degree of what we might refer to as focused attention. You're focused on me talking to you, you're focused on what's maybe shown up on the screen, right? And you're maintaining that attentional focus. That takes significant cognitive resources to do that, particularly for something that's goes on for an hour. But it's a very important aspect of how we interact with our environment. There's another kind of attention that's also important, and that is what's referred to as selective attention. And selective attention is referring to your ability to maintain attentional focus on potential salient cues in your environment, and to discard or ignore irrelevant factors or potential cues. And that might be if you're sitting here listening to me and watching the screen, and somebody's phone goes off and starts ringing, and it's not your phone, you know that that's not relevant to what's happening right now, and you hopefully will ignore that auditory stimulus and maintain attentional focus up here. And that's a form of selective attention, where you're selecting your attentional resources on information in your environment that you think is important or relevant to you, okay? So we all do that every day in our normal lives, and we are able to test those two kinds of attention in these rats. And the figure on the left shows on the x-axis, these are just for animals that did not get any Ritalin, and the numbers 1, 2, 3, 4, 5, and 6 are blocks of testing days. And so each of those blocks represents 3 days of testing. So in this task, the animals were tested for 18 days, 6 blocks of 3 days each. And you can see the white circles are the control animals, and you can see they gradually improve in their attentional accuracy in this focused attention task over those 18 days. The manganese animals do not. This is clear evidence of a deficit in focused attention in the manganese animals. And in the even more difficult task, to test selective attention, where we present them with the olfactory odor distractors, you can see that on the x-axis for the trials that had no odor distractor, they're doing pretty well, but again, the controls are doing better than the animals who got manganese. When we give them a distractor that's either 1, 2, or 3 seconds before the visual cue shown, you can see their performance drops substantially because they're being distracted by the visual cue. I'm sorry, by the olfactory, the non-salient cue. And both the control animals and the manganese animals are distracted by that olfactory distractor. The manganese animals more so than the controls, but they're all affected. Okay, that tells us that this task is working as it should. We're evaluating a form of selective attention in this case. Now, when we treat these animals with methylphenidate, we saw something very interesting. First off, if you look at the figure on the left, looking at the focus attention, we see that over the 18 days of the task, where the animals are getting Ritalin every day, It's not until at the very end of the task, in the last several days, that the control animals and the manganese-exposed animals on Ritalin are now performing identically. And so that tells us that the methylphenidate, the Ritalin that we gave them orally every day, had an effect, although it took some time, to alleviate the focus attention deficit in the manganese animals. And it was very interesting that it didn't happen right away, that it took 18 days, and this will be important in a few minutes. It tells us that in this case, that the benefit of the Ritalin is not due to the pharmacologic action of the drug in the brain at that moment in time. It's telling us likely that there has been some neurodevelopmental response to the daily dosing of methylphenidate, some neurobiological response that over time took a number of days to develop, and it was that response to the drug that is now responsible for alleviating the symptoms due to manganese. The figure on the right shows the selective attention task, and you can see now they enter this task right after the one on the left, so 18 days of the one on the left, and they immediately go to the one on the right for another 12 days. And you can see here the same thing, that in the 0 dose of Ritalin, manganese animals are performing more poorly in their selective attention compared to control animals. But when they get the lowest dose of Ritalin, they're identical. And that low dose of Ritalin completely alleviated the selective attention deficits in the manganese animals, right? And in fact, it was more effective at doing that than even the higher doses. And that's not an uncommon thing to see a dose response to drugs. And oftentimes we refer to that dose response, can express it as either a U-shaped dose response like I showed with manganese earlier, or sometimes an inverted dose response, U-shaped dose response. And that's what we see here. We see that the lower dose of Ritalin was the most beneficial to alleviating the attention deficits in the manganese animals, more so than the higher dose. And that dose response curve in the manganese exposed animals followed an inverted U-shape dose response curve, which is shown here, whereas the dose response curve in the control animals followed a regular U-shaped dose response. So that's important for a number of reasons, aside from just alleviating the attention deficit symptoms in these animals. It also tells us there's something different in the brains between the manganese exposed animals and the controls, because they're responding to the same dose of drug in a very different way. So I've just told you all this. I think at the end of this, we've come to characterize that this is probably the most well-defined animal model of attention deficit hyperactivity disorder that's yet been published. And we think that due to the developmental manganese exposure, that it really is contributing to a particular form of ADHD, and that's the inattentive subtype of ADHD. And we think that having this animal model now positions us very strongly to be able to ask questions about how the exposure early in their life is causing these symptoms. So I mentioned to you in the beginning that what is understood about ADHD in children is that it's believed to arise due to deficits in the functioning of the catecholamine system, that be dopamine and norepinephrine, in the prefrontal cortex of the brain, okay? And that hypofunctioning of that catecholamine neurological system is responsible for the symptoms that ADHD children often exhibit. So we did a bunch of different things. I'm not going to go through all that for you, but we looked at the amount of neurotransmitter being released in the animals' brains and saw that the manganese-exposed animals, who were only exposed for their first 3 weeks of their life, and we tested the release of dopamine in their brains and norepinephrine as adults, that they were releasing far less dopamine and norepinephrine compared to control animals. So that's the hypofunctioning catecholaminergic system. We found that these animals were expressing different levels of a number of different proteins and neurotransmitter receptors in their brain. The receptors that bind dopamine, like the dopamine 1 receptor and the dopamine 2 receptor, the proteins that are responsible for making dopamine, the proteins that are responsible for transporting dopamine back up into the cell, all those were at different levels in the animals who got manganese early in their life compared to controls. We also looked at expression of a number of different genes that I'll talk a little bit more about, that we think helps shed light on what really is causing these symptoms in the manganese-exposed animals. And most interestingly, because we are, whenever, let me just say, whenever you look at aspects of the tissue in the brains, of an animal model, it gives you tremendous insights on what may be changing at the neurobiological level. But there's also some downsides of those kinds of approaches because it's not the intact animal that's actually undergone the behavioral outcomes, where you can try to look at how the animal's altered behavior can be explained by things, altered brain function in the animal that's still functioning. And so to do those kinds of tests, we utilize a number of different things that are often used in people, imaging techniques used in people. One of them was positron emission tomography, PET scanning. We collaborate with folks up at a center at UC Davis to do that. We are also able to use something referred to as diffusion tensor imaging, DTI, to look at the diffusion of water in a living animal. And we're also able to looking at the connectivity of different neural circuits using the functional MRI. And I'm going to talk about a few of those things, not all of them right now. So for the PET imaging, this is a case where the animal is put inside a PET scanner, the live animal, that's lightly anesthetized, and we administer them through a catheter a radioactive compound. In this case, the compound was raclopride. And raclopride is a molecule that binds specifically to one of the dopamine receptors, the dopamine 2 receptor. And we can use that to actually probe in the living animal the number of functional dopamine 2 receptors that are expressed on the outside of their neurons. And shown on the left here is what one of these PET images look like of the rat's brains. And we can take the different intensities that are measured of the raclopride and how long it stays in that part of the brain over the scan, which takes about an hour. And from that, we can calculate the relative density or the amounts of these dopamine 2 receptors. And so we did that, and we didn't find any difference in the dopamine 2 receptors in these brains in the control animals compared to the manganese animals. And so that wasn't helpful in trying to elucidate what may be underlying the symptoms that these animals had. But then we also challenged the animals with a methylphenidate dose while they're in the scanner, meaning we gave them an injection of the Ritalin while they're in the scanner. And the Ritalin causes an increase in extracellular dopamine. That's how it works pharmacologically. And that increase in extracellular dopamine then would compete with the raclopride that's binding to the dopamine 2 receptor, and it would displace it. And so we could look at the rate at which the raclopride is displaced from the dopamine 2 receptor as a function of the Ritalin dose, and that will tell us how much dopamine was being released in response to that Ritalin dose they got. Well, so I mentioned we didn't detect any differences in the amount of those dopamine 2 receptors in the animals' brains in the control or the manganese-exposed animals. And you can see the age of these animals in the lower right. These were animals that were over a year old. And keep in mind, they got exposed to manganese for the first 3 weeks of their life, and now they're a year old. And a year old in a rat is equivalent to middle age in a human. Right? So it's quite a long time between the exposure and when this measurement occurred. So what we found when we challenged these animals with methylphenidate is that the manganese animals who got exposed, again, only the first 3 weeks of their life, that they actually released more dopamine in response to the Ritalin than the control animals did. And so we sort of had this like contradiction that we think, on one hand, we think the manganese and the ADHD is associated with lower levels of dopamine and dopamine function, but here we're seeing now that they're actually releasing more dopamine in response to that Ritalin drug. And what it actually tells us is it tells us exactly what we sort of uncovered in terms of the behavioral data, and that the manganese animals are responding to the drug completely differently than the control animals, and they're responding to those low levels of drug, which are shown to be the most beneficial for the behavioral, alleviating the behavioral symptoms, because they're actually releasing more dopamine in response to that drug than they should otherwise do. And so this is what we would think of as a compensatory or a compensating kind of neurobiological response that the manganese animals were showing. So we then subjected the animals to this diffusion tensor imaging. And this again goes into a scanner, and the animal's lightly anesthetized. And what diffusion tensor imaging does is it essentially measures the diffusion of water in the tissue, the brain. And so we had an undergraduate student, research volunteer, sit down with all the anatomical MRI images from all of our animals and go through section by section and assign a brain region to each of the anatomical images so we could distinguish the motor cortex from the prefrontal cortex, from the sensory motor cortex, and so on. And she identified 13 different brain areas that we are interested in. And then we looked at the data from the diffusion tensor imaging. And again, it is measuring the diffusion of water. And so we might think of water diffusing in a substrate like your brain. If there were no structures, water would diffuse in all directions equally. We'd call that isotropic diffusion. But if the brain, of course, we know has a lot of structures. It has cells, it has membranes, the neurons have axons, and those membranes restrict the extent that water can diffuse in given directions. And so normally water would be diffusing, or the intercellular fluid would be diffusing down along the nerve structures, so in a particular direction, and not just in every direction equally. And so we call that anisotropic diffusion. So in people that have had DTI measurements done, and in animal models, it's well established that if there's brain injury, say due to stroke, or an animal model of a stroke model, that increases the diffusion of water in all directions because there's damaged tissue that's damaged the structures of the brain. And they've also shown in a very few number of studies that if they look long after, months or years after an injury, that actually the DTI indices about diffusion actually increase, that there becomes more directional diffusion compared to animals or human that didn't have brain injury. And that's reflecting the fact that there's been neurological, essentially healing and regeneration of some structures in response to that injury. So in these manganese animals, we found, interestingly, that they showed increased evidence that the water was diffusing in specific directions, which is consistent with more well-established nerve tracts in the manganese animals compared to the controls. And that they also had reduced diffusion in all directions. And interestingly, the only brain areas where we saw that there was a difference in how the water was diffusing in these manganese animals were the brain areas that map to the deficits that we measured. The CG1 you see here, and the CG2, that stands for the cingulate cortex, 1 and 2. That's the prefrontal cortex. That's the brain area that's responsible for mediating attentional function. The dorsal striatum is part of that circuit, that loop, and the ventral tegmental area is also part of that circuit, shown here in the rat's brain, all this. Of all the 13 brain areas that we measured, only those show differences in the fractional anisotropic diffusion index. And when we looked at the mean diffusivity index, meaning that the water diffusion going in all directions, you see the manganese animals have a lower mean diffusivity index, meaning that it's not just diffusing equally in all directions compared to the controls. And that occurred in the brain areas only in the motor cortex and the sensory cortex, which are brain areas that are mediating the fine motor function deficits that we measure in the animals. So on the surface, people have looked at findings like this and said, this seems to be the opposite of what I'd expect. I'd expect that the manganese animals who had that high exposure during development and had a neurological insult, that they'd show evidence of damage. But we know that when there's early developmental insults, that there are compensatory healing processes and regeneration of different structures in the brain that actually map to what appears to be improved functioning. Now we know this is only diffusion of water, so this is only reflecting structural changes in the brain, not functional changes per se. And so for the functional changes, we looked, and this is the last piece of information I'm going to show you, we looked at the methylation of different genes in the prefrontal cortex tissue, and we looked at the expression of different genes in the prefrontal cortex tissue. Of course, there were thousands of genes that we saw that were differentially methylated or changed. And some of you may know that methylation, which is a C carbon with 3 hydrogens, a methyl group, that DNA can be methylated when having one of those methyl groups attached, and the degree that those methyl groups are attached can affect whether that gene is expressed or not. And so we know that genes their level of expression oftentimes is associated with the translation of those transcripts into functional proteins. And so we were particularly interested in looking at genes that we knew were related to the function of dopamine circuits within the brain. But mostly we were trying to understand what was it that was really causing all the changes that we were seeing that would result in the behavioral symptoms that we were measuring. So on this slide, they're just things you don't need to even try to read or whatever. I'll tell you that one of the things that we identified as one of the top causal factors that could explain the expression changes of many of the genes that we measured was this protein called mammalian target of rapamycin, mTOR. This is something that's been implicated in a whole variety of different biological functions and diseases, including cancer. And it's almost ubiquitous in terms of what people seem to think it's related to regulating. But for us, we recognize that it's upstream and is a key regulator in determining how much of the dopamine receptors get expressed on the neuron surface, how much dopamine gets made, synthesized, and released. And so it's well established that it plays an integral role in regulating catecholamine or dopamine and norepinephrine function. So we related the changes in this mTOR, shown in the middle of this spoke diagram, and actually many of the neurological function genes that were shown to be changed due to manganese exposure back in the first 3 weeks of their life, many of those genes that were shown to be changed in these animals' brains that got manganese, those expression of those genes is controlled by this mTOR. So we think that it plays a very important role in how it may help explain the neurobiological changes that we see, and then hence the behavioral changes that we see. Now, this is an extremely complex diagram that took one of my graduate students about a month and a half to make. And I'm not going to go over it with you because it's rather tedious. But I will tell you, if you look at the panel on the left, the summary, we're able to put together the pieces, we think, with the data that we have that helps us understand how an early life insult to elevated manganese can produce the neurological damage, that can produce the altered neurological function, that then produces the behavioral symptoms that map perfectly to ADHD. So at the top, we have the manganese insult. Elevated manganese during a life stage, developmental life stage where the brain's developing, very sensitive, and where the intake and retention of the manganese is high in these young children. Manganese under normal levels is essential, I mentioned that, but at high levels, it can actually be a very powerful pro-oxidant, can cause oxidative stress, right? Some of you may have heard that. We all know what antioxidants are, and we take antioxidants to help guard against things that are pro-oxidants. And manganese is well established as to cause increased oxidative stress early at high exposures during development. So we think that this early life insult, increased cellular injury and oxidative stress, that's the first up arrow. The normal response to cell injury and oxidative stress is to increase pro-inflammatory response, upregulate inflammation. We all know that inflammation, chronic inflammation is bad. Acute, I tell my students, acute inflammation is good because acute inflammation resolves itself. But if it's chronic inflammation, it doesn't. And we think this early life insult with manganese generated this chronic inflammatory condition in the brain. This inflammation is well established, and I'm going to skip a couple layers to affect a number of different signaling proteins that we have evidence were changed in these manganese animals with the QKI signaling and the Wnt signaling and so forth. At the end of, or near the end of this, is altered mTOR signaling. And the mTOR signaling then is directly related to the reduction in the dopamine receptor, dopamine 1 receptor protein expression levels, in the reduction of dopamine production, and in the increase in dopamine 2 receptor levels. And we saw both those things in the animals. Some receptors were downregulated and others were upregulated. And we think and believe that it's this altered expression of these receptors due to this altered mTOR regulation by manganese and oxidative stress that actually sets the cascade in motion that results in the neurobiological damage that then produces the symptoms that manifest as ADHD. Now, I'd like to think that we were the first ones to come up with this. But just coincidentally, there was a review paper that was posing a hypothesis. And this review paper, which I show here, was posing that this signaling pathway, not in the same way that we thought about it, but it was posing this signaling pathway was instrumental in the development of attention deficit hyperactivity disorder. And the signaling pathway has been shown to be altered by chronic dosing with Ritalin. And so we're now having the convergence of some initial evidence in the epidemiological literature with the animal model work that we've done that now is coming together to help us better understand what is it that's causing ADHD. And we know that there are many ways that a child can acquire a disorder like ADHD. There's not just one way, but now we know that environmental insults that can cause injury to the brain during early development, like elevated manganese exposure, there are likely countless other kinds of insults that could also cause this, could during development, set them on a path for being at a much higher risk for developing the altered neurobiological function, and then, of course, the symptomology that's associated with ADHD in this case. And we think that this approach and understanding environmental contributors to behavioral disorders likely also fits quite well with other behavioral disorders, including autism and others, because they're all sort of under an umbrella of altered neurobiological function that in which the symptoms can be arrived at likely via multiple pathways of insult early in life. Okay, so that's it. I think I have a summary here that just refers to everything I just said. You don't need to read that. I think what I want to do is point to some of the champions that have really enabled this work, and this is a poster session we did at the university last year, and there are many of the undergraduate students and some graduate students that were instrumental in helping us put this body of work together so I could talk to you today. And with that, I'm going to stop and take any questions. Yeah.
Okay, we have time for a couple of questions. Constantine, and Anandi has the microphone here.
So we've established that manganese is one of the culprits of causing this. And so the next step is, how do we get that manganese? And has anybody done any research on the foods that we eat that we shouldn't or should?
So I think it's an important point to clarify. So if we all ate our natural diet, including as infants being fed largely exclusively through breast milk, this would be a non-issue, obviously. It's only, I think it's only in terms of our diet, it's only when there's an excess of things that we logically would not otherwise do. There's high levels of manganese naturally in some teas, high level in soy products, for example. But again, for anybody over the age of, young adolescent, it's absolutely a non-issue. There is an interesting part to that question that I think it's worth mentioning, because we think of, in the field of toxicology, we think of everything that's got to do with exposure. But we realize that this is very important, that we're not all the same. We're all, there are very important differences. And we have data in this Italian cohort of children I mentioned to you, that there's genetic differences in some of the genes that are responsible for making proteins that transport manganese. There's genetic polymorphisms in these that make those individuals more or less susceptible to having higher or lower body manganese levels naturally. And so you can imagine now, this is a classic environment by gene interaction, that if you are somebody who has one of these polymorphisms that makes you less able to excrete excess manganese, then you'll be more inclined to retain more, have higher blood and body manganese levels, and therefore be even more susceptible if you have higher exposures.
Is there a correlation between breastfeeding and ADHD?
So there was one of the fellows, this is a long-winded answer to that excellent question. One of the fellows who got us interested in this work was a clinical psychologist at UC Irvine. And he had that theory that children who were not breastfed, but were fed formulas, We're more likely to, he was a clinical behavioral psychologist, more likely to have academic difficulties, maybe be diagnosed with ADHD, maybe higher propensity for criminal activity as an adult. There have been studies looking at infant formulas, but there has not been a proper epidemiological study looking at whether children that are fed exclusively off bovine or soy-based infant formulas versus breastfed children have a higher risk. And we've often wondered why that's the case, why it hasn't happened.
All right, we're running out of time, but one last question here.
I have a question about heavy metals, other heavy metals that affect the brain. And as an expert toxicologist, you know that mercury, one of the most toxic heavy metals, is in all flu shots and has been associated with autism, although they have never proved it. There's research on both sides proving it and then disproving it. And autism also has a symptom of ADHD. And in California, 1 out of 22 kids are now autistic. There's also aluminum in most other vaccines, which has been classified as a heavy metal and is associated with Alzheimer's. And if these metals found in vaccines could be eliminated, as well as manganese in soy milk, wouldn't that be more effective than treating it with some kind of drug? Because the brain still gets damaged from the toxins.
So that's an excellent set of questions. Thank you. And there's several questions in there that I want to respond to. The first, which is related to one of the last things you said, is super important. And that is that primary prevention is the only way to, in my opinion, to effectively treat these kinds of disorders. And primary prevention is eliminating the exposure before it occurs. And so we've learned, I used to study lead and lead poisoning for many, many years. We've made enormous strides and have enormous successes in lead poisoning and the frequency of lead poisoning in children by measures of primary prevention, right? By eliminating exposures. We all know, I'm sure all of us know about that. When it comes to other things like the mercury and the thimerosal, the vaccines that you mentioned, aluminum and Alzheimer's, what makes it so difficult to understand in those kinds of cases is to establish that there really is or not a causal relationship between the exposure and the outcome, like autism. And that's the challenge with epidemiological studies in general, is because by the time a child is recognized and presents or is diagnosed with a behavioral disorder, the thing that may have contributed to that disorder, or the things, may have occurred years, if not decades before. And the extent to which studies are able to accurately measure or estimate or model those things varies substantially. So I think that's one reason why it becomes so difficult to actually establish those linkages. So I do want to underscore the primary prevention is clearly the thing that should be focused on for all these toxic metals, including mercury and lead. Manganese is a little bit more challenging because it's also an essential metal. But for the ones that have no biological function, like mercury and lead, absolutely, I think that should be the focus.
Okay, well, thank you very much, Don. It's really great work. So we hope to see you in September. Many of the interest groups keep going over the summer, so stay with OLLI, read your notices, and come to the picnic. Come to the picnic in June. I'm sure Don is open to questions if you want to come up.
Both of the